Hello, everyone. Thank you for attending today's webinar, How Continuous Safety Monitoring Can Reduce Hospital Stays for High-Risk Oncology Patients, presented by Fierce Pharma and sponsored by Current Health, a Best Buy Health company. I'm Katie Green, the Associate Director of Marketing at Current Health, and I'll be the moderator for this session. I'd like to start off by introducing today's speakers. We have with us Dr. Sridevi Rajiv, the Assistant Attending Physician at Memorial Sloan Kettering Cancer Center, and Dr. Nicole Zaradka, Senior Clinical Research Program Manager at Current Health. You can read their full bios on the left side of your window by selecting the Speakers tab. Quick look at today's agenda. We are going to start off with an overview of CAR-T therapy to give us some context. Then we'll dive into the research study that assessed the effects of continuous monitoring. And finally, we'll zoom back out and talk about the financial implications of this research. Just a few quick notes before we begin. You can access closed captions from the bottom right corner of the video player. This webinar is being recorded and will be available to watch on demand in the next 24 hours. Finally, we'd love to hear from you. During the presentation, please submit any questions you have using the Questions and Answers tab on the left side of your screen. We do plan to cover a few questions at the end of this session, and we'll get back to you personally if we don't have time to get through all of them, so we appreciate your patience. With that, let's begin. Dr. Rajiv, I'll turn it over to you.
Thank you for the introduction, Katie. My name is Dr. Sridevi Rajiv, and I'm very glad to be here talking about some of the interesting research that we have done so far and the results of our investigator-initiated trial. But before I step into the results of our research, I just wanted to give our listeners some background onto CAR-T therapy. So I just want to start by providing a very brief overview of the latest immunotherapy approach we have for hematological malignancies, also known as blood cancers, called CAR-T therapy. Now, CAR-T stands for chimeric antigen receptor T cells. These are white blood cells, which are the immune cells of our body, which fight both infection and cancer, called T lymphocytes. These T lymphocytes are selectively collected from a patient, modified in a lab, and I will go into this process in the next couple of slides. And these modified cells are given back to the patient, and these are called CAR-T therapy. Now, when the patient receives the modified T cells back into their body, these CAR-T cells start expanding in the body and start to elicit their anti-cancer activity. This sort of triggers a cytokine storm, triggering an adverse reaction called cytokine release syndrome, henceforth abbreviated as CRS. CRS, we have noted, occurs in almost 50% to 90% of patients receiving all kinds of CAR-T therapy. Now, across multiple myeloma, lymphoma, and leukemia, there are around 5 to 6 approved CAR-T products and multiple experimental products. And what we have seen is that the timing of the onset of cytokine release syndrome, or CRS, completely varies between these different products. CRS is often graded from grade 1, being the mildest, to grade 5, which is quite severe, leading up to death. Grade 1 involves only fever. Grade 2 involves in fever with low oxygen saturation or low blood pressure. And as we go up on the grades of CRS, there is a more severe low blood pressure and severe low oxygen levels that are happening to the patient. Now, cytokine release syndrome often tends to start as grade 1, and if not intervened at the right time, they can quickly escalate to grade 3, grade 4, or even grade 5. And this escalation can happen over a matter of mere hours, anywhere between 3 to 18 hours. We have often found in our hospital administration and QI projects that higher grades often require more intervention, and these interventions directly translate into more healthcare utilization costs up to the range of $100,000 to $200,000 per patient. We intervene to mitigate CRS by an anti-interleukin-6 monoclonal antibody called tocilizumab, or another drug called anakinra or corticosteroids. The administration of these drugs significantly reduces the severity and the duration of cytokine release syndrome. Next, please. As promised, here's a very brief overview of the CAR-T infusion process. If you look at the cartoon here, you can see a man who is sitting in that chair, and this is very similar to a patient we have in our apheresis unit. Now, apheresis is the process wherein the white blood cells are selectively collected using large cannulae from the patient. We take it to the lab where these T cells are separated and removed, and they are genetically altered to have some special receptors called chimeric antigen receptors expressed on the surface of cells. These cells are then made to multiply in logarithmic accounts to express these receptors on their surfaces. These multiplied cells are now armored with these new receptors that can selectively bind to the cancer cells, and they are reinfused back into the patient. The whole process, right from collection of these white blood cells back to reinfusion after being processed in the lab, can take anywhere close to 3 to 4 weeks if there are no delays anticipated. Next, please. Currently, the standard of care of how we administer CAR T cells is that patients get admitted to the hospital. They get 5 days of a conditioning chemotherapy, what we call lymphodepleting chemotherapy. And after from the start of lymphodepleting chemotherapy, after 5 days, we infuse these CAR T cells back to the patient. From the time of infusion of the CAR T cell, in the vast majority of hospitals across America, across North America and Europe, patients stay in the hospital anywhere between 7 to 14 days, whether it's a commercial product or an experimental product. And this is where we identified that there might be a problem that we have to solve. Because patients stay in the hospital for anywhere between 7 to 14 days, these prolonged hospital stays translated into increased healthcare utilization costs, reduced availability of hospital beds for patients who have acute problems, increased infection risks, and overall patient dissatisfaction. So we decided to design an investigator-initiated clinical trial in which we looked at patients who were admitted to the hospital getting CAR T therapy and adding on a layer of monitoring using wearable device technology for the duration of their admission. And the purpose of this investigator-initiated trial was to see the feasibility of this wearable device effectively capturing vital signs and comparing them with standard of care, which is nursing recorded vital signs, and trying to see if the wearable device technology can efficiently capture vital signs as well as nursing does. So this is a rough schema of what our trial looked like. Patients got admitted 5 days ago, and on day 0, which was the day of CAR T infusion, patients who were enrolled in this trial also started wearing the wearable device, and they wore up to their full period of monitoring while in the hospital. Now, we have written CAR T product A, the high-risk period being from day 0 to day 4, and that is, according to what I had mentioned before, certain CAR T products have an earlier onset of cytokine release syndrome, so we are on high alert for that particular product in the first few days. And on the other hand, as exemplified by CAR T product B, the high-risk period tends to be much later in the course of that admission. So the patient is essentially staying in the hospital for 7 days after CAR T infusion without any vital sign changes, but it's towards day 8 and day 10 that we start seeing symptoms of CRS. So the problem that we try to tackle is how can we reduce this whole inpatient stay for patients with a manner to effectively monitor their vital signs. Next slide. Again, with the commercialization of CAR T products all the way back from 2016 to 17, there is a huge push to try to move CAR T therapies and a newer model of immunotherapies called bispecific antibody therapies to an outpatient infusion and monitoring setting. Because the current standard of care is that patients get admitted for CAR T therapy for the duration of monitoring period and for bispecific antibodies for the first step-up dosing. There are many advantages to trying to reduce the inpatient stays. It reduces patient burden, caregiver burden, it reduces infection risks that are inherent to an inpatient stay, it reduces bed occupancy, which is a huge problem in tertiary hospitals, and it can overall reduce healthcare associated costs, both direct and indirect. However, moving these therapies to the outpatient setting is not as easy as it sounds. There are many challenges and risks that we have identified. Some of them include the absolute need for close monitoring for cytokine release syndrome, the fact that there's a huge unpredictability of clinical course, meaning at this time, if I'm admitting a patient for CAR T, there's no way for me to predict whether this patient would be at a higher risk to develop CRS, or whether if this patient is at a higher risk to develop a higher grade CRS. There's no prediction method whatsoever at this time. There's also no objective risk-benefit assessment for patients who would benefit from this prolonged monitoring versus abbreviated monitoring. And finally, and this is something that's evolving in 2023, there's a reimbursement uncertainty because the reimbursements for these costly therapies are right now all built for an inpatient setting. So in our investigator-initiated trial, we tried tackling these problems in two different ways. For the need for close monitoring of cytokine release syndrome, one of the main adverse effects of CAR-T, we decided to add on wearable technology for remote patient monitoring and comparing how effectively they perform in capturing cytokine release syndrome. To try to predict whether which patient would be at a higher risk to develop CRS, or if they're at a higher risk to develop higher grade CRS, we are studying immune correlates, which are nothing but biomarkers or cytokines to help predict cytokine release syndrome. For the purposes of this webinar, I will be only talking about the wearable device portion of our IIT. Next, please.
So the goal of this work was really to address the question of, can technology help us solve these challenges? And as Dr. Rajiv described, we did this in twofold. We looked at the feasibility, so will patients adhere to the monitoring? And we looked at the time to detection of cytokine release syndrome. Is earlier detection possible? To address this question, we conducted this observational study at Mount Sinai to collect wearable data while patients received standard of care monitoring following CAR-T infusion. And what this enabled was a direct comparison of CRS detection and time to detection within each patient. The device selection in this study needed to include vital signs of importance for CRS detection, such as temperature, but it also needed to be equipment that clinicians felt confident in when we translate this work into the outpatient setting. Current Health is a platform designed to monitor high acuity patients at home. It includes an FDA-cleared wireless sensor that continuously and passively monitors several vital signs. Additional devices can be integrated into the setup based on patient and program need. And the devices are pre-configured with Wi-Fi and paired, so the data is seamlessly transmitted to healthcare provider-facing dashboard. And this is where healthcare providers can view patient data in real time. This data can also be stored in the patient's medical record through EHR integration capabilities. And so because of that, we've selected both a wearable and both the Current Health wearable and this temperature patch to simultaneously collect data while patients were receiving standard of care monitoring. So here's the study schema, and as Dr. Rajiv described, patients are inpatient, they are receiving standard of care, and we start the remote patient monitoring, so the data collection in parallel to standard of care on day 0 through discharge. Okay, and what this means is that the data are collected, but clinical decisions are not made from the Current Health data at this point. This is a retrospective data analysis. And again, I just want to highlight that because we are collecting these two streams of data in parallel, it allows for this direct, rigorous comparison between time to detection of CRS, which is not possible in an outpatient setting to do such a direct comparison. To evaluate feasibility, we assessed adherence during the monitoring period as well as during the high-risk period. And as Dr. Rajiv described, the high-risk period is the window of time post-infusion that has the highest likelihood for the development of CRS, and that was reported in the literature. What we found was that wearable adherence was 68% over the monitoring period and 73% during the high-risk period. And this adherence is related to data quality. When wearable adherence is high, like in this work, we know that we're generating data, and we know that that data are of high quality and exist to enable, this post collection analysis. And this is really important for us to be able to accomplish the goal of CRS detection. One of the ways that we analyze this data retrospectively, which is after it was collected, is looking at the onset of CRS using visual inspection. And this is where two clinicians independently mark where CRS begins in the vital sign plots. And so what you'll see is this figure on the right is an example of the 4 vital signs that were collected in one patient over their monitoring, over a few hours of their monitoring window. And on the vital sign on the top left plot is oxygen saturation, and then going clockwise, we have pulse rate, temperature collected through the temperature patch as well as through the current health wearable. The skin temperature is the one that's collected through the current health wearable. And then the last vital sign that we look at is respiratory rate. Now, the vertical lines that you'll see are the clinician tags, so where the clinicians observe the onset of change in vital signs indicative of CRS, as well as the timestamp of when the nurse recorded vital signs indicated CRS onset. One of the things I'd like to note, in this example, you can see it, but overall, the inter-observer correlation for this work was 0.76. And what the inter-observer correlation represents is the agreement between 2 independent observers, where one is a perfect correlation. Now, Dr. Rajiv, I'd love to get sort of your experience as one of the clinicians tagging this data set. What about this data, when you looked at it, excited or surprised you once you got into it?
Yeah, absolutely. You know, again, just taking these 4 graphs as an example, as I was annotating, and again, this was a blinded annotation. I did one set myself, the other was done by Dr. Wilkes, was part of Current Health. And the we started out was that solid black line, which was the point in which we know that nurses recorded cytokine release syndrome. And then as I was looking at this data, I started seeing this difference in vital signs started way before nursing recorded CRS was actually done. So that was pretty exciting for me. Honestly, when we started out the study, what we had in mind was that, is the wearable device good enough to be comparable to nursing recorded vital sign, meaning is it a non-inferior, or what we call in medicine a non-inferiority study. But as the data came in, we started realizing that it is not only not inferior, but in fact it's actually potentially even superior to nursing recorded cytokine release syndrome time points, because as you can see, there is an earlier capture of changes in temperature, there's an earlier capture of drops in oxygen saturation, there's an earlier capture of variations in heart rate as well as respiratory rate. And that was completely surprising for me. I was not expecting this much of an earlier capture, or what I like to call as a lead time, from when we initially start detecting changes consistent with CRS and when we actually call it CRS by nursing. So that was quite pleasantly surprising to me.
Amazing. And now, these visual inspections are really useful in demonstrating that these trends do exist in the vital signs for the historical data set. But in addition to this, we wanted to explore methods that would enable this real-time detection of change that was scalable. Next slide. And so we evaluated 2 threshold detection methods. And what that means is that we created a value, that's the threshold, and we said when the temperature crosses this threshold, that is when we're detecting CRS and using an automated method. Now, the first method that we used was a traditional method, okay? We used a value that was the traditional definition of clinical grade fever, 38 degrees Celsius. And then the second method we used was a personalized threshold. Now, this personalized threshold was created by using 2 standard deviations above the patient's mean baseline temperature. And the reason we selected 2 standard deviations above the patient's mean baseline temperature is because statistically, when you move 2 standard deviations away from the mean, you capture about 95% of the variance in your data. And so we made the assumption that when you're continuously collecting data, such as temperature, you're going to have natural fluctuations, especially in the home, especially during activities of daily living. And so we said, okay, if we create a personalized threshold that's 2 standard deviations above the mean baseline temperature, then when we cross it, we're probably experiencing a meaningful change. It's probably no longer noise. And so we have this graph on the left that represents the monitoring time on the x-axis and temperature on the y-axis. And the purple represents the temperature over time. And what you'll see are these horizontal lines. Again, the dotted line is the individualized or personalized threshold. The solid line is the fixed clinical definition of fever threshold, 38 degrees Celsius, and the triangle and the black vertical line indicates when the nurse recorded vitals indicated onset of CRS. And so we use this method to evaluate the time between the standard of care and these automated threshold methods in terms of detection. Here's some of the things we found. We found that the median personalized alarm threshold value was 37.4 degrees Celsius. This is a bit lower than that 38 degrees Celsius and that's, again, the clinical definition of fever. We also found that the wearable detected temperature changes consistent with CRS 180 minutes earlier than standard of care using that visual inspection method that I described previously, 46 minutes earlier than standard of care using that fixed alarm threshold of 38 degrees Celsius, and 206 minutes earlier than standard of care using that personalized threshold. And so what this really indicates is that these preliminary results show that the detection of CRS in the wearable data preceded the standard of care, and that was corroborated by all 3 of these methods. And the thing about this early detection is that enables early action. So Dr. Rajiv, can you speak a little bit to the utility of this early detection and how that might be useful in an outpatient setting?
Absolutely. As we discussed earlier, Nicole, this earlier detection was quite an unexpected outcome of our study. And so we're still grappling with the clinical implications we can play around with. So an earlier detection will allow us potentially, if we were to take these treatments to the outpatient setting, and let's say if we detect these an hour or 2 earlier than they would have been detected if they were in the hospital, that gives us clinicians the time to call patients, for our team to call patients, triage them, understand the circumstances behind these vital sign change, and make a determination if we are actually seeing clinical cytokine release syndrome or the start of cytokine release syndrome. And again, this lead time can be utilized for not only just triage, but if we were to bring them back into the hospital to give them pharmacologic treatments, again, all this time can be utilized in the transportation, admission to the hospital, etc. So I feel that this lead time is giving us a lot of buffer time to act before any grade of CRS happens or it worsens into a higher grade of CRS. So this is great, actually. It really, once validated, will help us, I believe, move these therapies to the outpatient setting. So switching gears a little bit away from the actual findings of our study, I just wanted to touch very briefly of the financial landscape of CAR-Ts that a lot of patients and a lot of clinicians are not quite aware of. Now, granted, CAR-T cell therapies are extremely efficacious for people who have seen multiple lines of therapies, but these are some of the most expensive drugs in oncology till date. For example, for an entire CAR T cell therapy for a patient, it can range anywhere between $500,000 to $1 million based on, their length of their admission, the complications they develop on their way, etc. Now, the cost of manufacturing a CAR T alone in the process that I explained quite a few slides before can range anywhere between $373,000 to $475,000. So that is a fixed cost that is out of the hands of the clinician. Now, based on the present model, when every single patient is admitted, as you can see from this graph that was adapted by a study that was led by Dr. Jagannath at Mount Sinai and some pharma partners, they modeled 3 different scenarios in which patients receiving CAR T cell therapies are admitted. So the first scenario was when there is a 100% inpatient admission for the whole duration of monitoring. The second scenario was when there was an 85% inpatient admission, followed by 15% outpatient monitoring. And the third scenario was bringing down the inpatient stay even further to 70% and observing the patient outside the clinic for 30% of the time. And again, the punchline of this entire graph is that when a patient has more inpatient stay, there is increased cost associated with inpatient stay, the peri-infusion costs, the nursing costs, the stay costs, etc. If we were to reduce the amount stayed inpatient and increase the time that you monitor outpatient, there is significant implications in reducing costs and more financial sense. Again, we have to do it all in a very safe and effective manner by not compromising on monitoring for these adverse effects, which is where I feel wearable technology can come in. So if a patient were to have adverse effects, you can see on the very last bar graph that the adverse effect management costs are exactly the same. But what is quite different is the peri-infusion cost, which varies between the level of administration of these drugs, which is inpatient versus a hybrid of inpatient or outpatient. Overall, the key takeaway from the paper was that the lesser time that you have, which is being monitored inpatient, the more financial gain that you may have by reducing costs. The global CAR T cell therapy market is poised to grow as high as 30% from 2023 to 2032, owing to a variety of factors, which can be attributed to the increasing incidence of cancers globally, the therapy being available to more patients because we have increased manufacturing, increased FDA approvals of these therapies, increased experimental products that are coming to the market. So overall, the demand and supply of CAR T cell therapies are increasing and only will continue to increase as the years go by. But this also poses a huge burden on hospitals if we were to keep admitting every single patient getting a commercial CAR T or an experimental CAR T product. So we have to find a way to optimize giving all these patients the treatment they need, but also reducing the reliance on complete inpatient monitoring and maybe just daring to think outside the box and having a portion of patients being monitored outpatient. So just to touch base very briefly regarding how many patients do end up getting cytokine release syndrome and how does that really look like across the different hematological malignancies and across different products, roughly 50 to 90% of patients undergoing this CAR T cell therapy get CRS. I can say that if we admit 10 patients, 8 or 9 out of 10 patients always get cytokine release syndrome, and mostly recently it's been lower grades, but we do have patients who still end up having grade 2 or above or severe grade CRS. This graph on the right is showcasing the costs associated with treating higher grade CRS, and you can see that as the grade increases, there is a doubling or even tripling of the costs associated with treating the cytokine release syndrome. There is another main adverse effect that we also monitor following CAR T therapy called neurotoxicity. Now, neurotoxicity is an entity that's completely separate from cytokine release syndrome. It can appear on its own independent of CRS, and it can also appear concurrently with CRS. So overall, taking into the adverse effects profile of a patient with both cytokine release syndrome and neurotoxicity, as the patient experiences a higher grade of cytokine release syndrome or a higher grade of neurotoxicity, the more drugs we have to give them, the more frequently we have to administer, we escalate their monitoring not from the general medical ward to the ICU setting. So all that translates into a higher cost incurred per patient for increased grade of adverse effects. And what this behooves is that if we were to intervene at an earlier grade, let's say we capture patients right when they have grade 1 cytokine release syndrome or right when they have grade 1 neurotoxicity, and we intervene right at that time, we can significantly bring down costs by not letting the cytokine release syndrome evolve into higher grades of severity. And this is where, again, I think the results of our IIT are quite promising. In addition to the data capture, we also looked at the sensitivity of data. We thankfully found that the device in patients who were completely adherent, which was quite significant, we found that there were no missed events. And in fact, the device even was able to capture subclinical events, meaning events that were not really called by our clinical team as CRS, but the device really picked up the changes. So we are calling them that the device is sensitive enough to capture subclinical information, and it's not missing any event, which is quite reassuring if I were to send my patient to the outpatient setting. Next slide, please. So the key takeaways from our study and in general, what we are attempting to do is that continuous vital signs monitoring can feasibly support an outpatient CAR-T therapy experience. Earlier detection of vital sign changes associated with cytokine release syndrome will help with timely triage and timely pharmacologic intervention, including upgrading the site of care from home to the hospital or from within the hospital in the inpatient setting to the ICU. By reducing or eliminating inpatient stays associated with CAR-T infusions can potentially bring substantial cost savings to healthcare systems and also to providers. As Nicole had very succinctly pointed out, individualized vital sign thresholds can showcase maximum sensitivity in detecting CRS, and these may pave the path for development of personalized predictive algorithms. At-home care following CAR-T infusions could provide better patient experiences and enable better accessibility of treatments, which are not just confined to large tertiary hospitals, and we may be able to take these life-saving treatments to even smaller centers and outpatient clinics.
This work really highlights the, again, direct comparison within patients to the utility of continuous monitoring through our current health wearable and time to detection of CRS. And then it becomes the question of how does this translate to the outpatient setting? Now some of our clients are already running these outpatient programs post CAR-T infusion. And unfortunately, Current Health has a lot of offerings that support programs like this. Some of the things that are included are our clinical command center, which can alleviate some of the burden that comes with continuous data collection and remote patient monitoring in the outpatient setting. What they do is in our current clients, they sort of bridge the gap for out of hours monitoring from the clinic. So think nights, weekends, etc. We have features available in our platform, such as patient self-reported vitals and symptoms and patient survey alarms, which, although weren't integrated into this particular research work, because that was sort of not the goal of this research work, could be really useful, again, monitoring patients in this outpatient setting. And lastly, we have logistics support, and how our clients are using that today is really program-based on need. And so some programs prefer to deliver our equipment in clinic in person. Some prefer to deliver in home by a clinician. And then some prefer to use our logistics support, which is at-home delivery, or some hybrid of this. And so we really have these offerings, again, to support the success of your program and to take this very complementary research work that we've just presented and translate into at-home care. Thank you. I think we have time for some questions. So Katie, are there any that have come in?
Yes, indeed. Let's move on to taking a few of these. Let's see. Our first question is, aren't new CAR T being developed with less risk of CRS? Is this still as beneficial? Dr. Rajiv, do you want to take that one?
Yeah, I can definitely take that one. That is a very insightful question that was asked by someone who knows some of the CAR T data. So I appreciate that question. So it is true. The older generation or the first generation CAR Ts used to have significantly higher CRS incidence as well as higher grades of CRS, meaning we saw much more grade 3 CRSs, grade 4 CRSs, and a lot of patients ending up in the ICU. Since the last couple of years, the products that are coming to the market are what we call the second generation CAR Ts, and also we have in development third and fourth generation CAR Ts, all of which are designed to reduce the risk of CRS itself. That being said, so far what we have seen is that the CRS risk is reduced in terms of severity by, we are no longer seeing these high incidences of grade 3 and grade 4, but we are still seeing an overall incidence of CRS as high as 80%. And even in the most advanced CAR T products in which they secrete cytokines to mitigate CRS from within the blood itself, we are still seeing a CRS incidence as high as 50%. So this is going to be a persistent problem. I think, we can expect cytokine release syndrome despite newer generation of CAR T products coming in. So that's all from a cell perspective. Now, patients who are going into CAR T therapy with a high disease burden, or patients who have what we call an immunosuppressive tumor microenvironment, they all are at higher risk for cytokine release syndrome, despite giving them second generation or third generation CAR T products. So we do expect CRS in brief in the vast majority of patients we give these treatments for. So it is quite necessary to monitor them very closely and continuously for CRS.
Fascinating. Our second question is, how do you see technology evolving to advance predictability? Dr. Zaratka, do you want to tackle that?
Sure. So let's break technology into two components. There's hardware and then the software side of it. To be completely honest, I don't think that the hardware side is really going to evolve. I think that the raw data that's coming out of those sensors are going to continue to come out of those sensors, and it's really the algorithm development that's going to improve. And so I think that from the state of threshold alarming, we really have a opportunity to create some more sophisticated algorithms for detection without having to change the hardware. In addition to that, I think the addition of forecasting vitals, as well as adding some other inputs to predictive modeling, whether that's biomarkers or demographics, et cetera, will really enhance the predictability of cytokine release syndrome in the future.
Our third question, let's see. Could you share any feedback from patients who have undergone remote monitoring during their CAR T therapy?
Sure. As part of the investigator-initiated trial, I was fortunate enough to run at my training hospital at Mount Sinai. Patients, when we approached them with this idea, they found the idea quite novel, and they were always interested to hear more. So whenever me and my team approached them, we approached them not just with the concept, but also by showing them the wearable, which is actually just a little bit bigger than a smartwatch that most of us do wear. So initially, when we talked to them, they were thinking that it's going to be a big, cumbersome band to wear. But when we actually showed them the sensor, they were like, oh, if it is this small, I can definitely wear it. And when they started wearing it in the hospital, I think the vast majority of patients did not have any issues wearing it. I think the biggest problem we faced was that sometimes their stay in the hospital was so uneventful until that cytokine release syndrome actually kicked in that they often forgot to charge their device. This device can hold charge for up to 24 hours. So sometimes we see that the flow of data just stops, and we instantly know by looking at the platform that their batteries at a 0%, and these people have kind of forgotten to charge despite us reminding them every day. So what we did was that a representative from our team used to go and remind these patients to charge, and to make uniform across all patients. We were like, at lunchtime for 1 hour, just keep your device for charging each day. And that kind of significantly improved this whole issue of forgetting to charge and placing the device back on. But overall, there were no complaints of any discomfort. There was no complaints of any skin irritation. There was even one patient that went to the ICU wearing the device, and we did not receive any reports that it interfered with any of the machines that were used for intervention or monitoring.
I'd love to piggyback off that a little bit, especially about the charge and the charge status. So one of the available opportunities in the outpatient setting is using a tablet tied to, like, as part of the kit. And what this really allows the clients operating these outpatient programs to do is set reminders for charge, and sort of enable this engagement with the patient in an automated fashion, that, again, because of the way that we had this particular research study set up, wasn't available to the patients in the inpatient setting. But with the at-home kit, there is a tablet. You can set reminders. There is a little more automated feedback that they receive. And so we are seeing this event of forgetting to charge the device a little less in those groups.
Well, I think patients will love that if, they use their tablets properly. Because the study was done inpatient, we did not use the tablet portion that's being offered by Current Health. But I think if we were to take it to the outpatient, that is something that I will definitely be using for sure.
Always so interesting to see how people's behavior changes in different contexts. We've got time for one more, and it's kind of a multi-part one, so bear with me. Where does this research lead you to your next endeavor? What questions need to be answered by research? And how will this model evolve as personalized immunotherapy advances? I like both of you to weigh in on that.
Yeah, absolutely. I can go first because I think from what I heard, all these three questions are overall asking about what's the future, after the study. What are we dealing with, and how do we envision this to change in the future? Now, I can tell you my bit that the results of these study were quite, hypothesis confirming as well as hypothesis generating for me. So what I would like to see is to take these results from a single institution, single arm study to a wider or a larger sort of phase 2 trial setting, which is multi-institutional and also drug agnostic. And what I mean by drug agnostic is that not just include patients getting CAR T cells, but also include patients receiving bispecific antibodies, which are another class of novel immunotherapy agents that are also known to cause cytokine release syndrome. So I would want to validate our findings across different institutions, different settings, different drug products that elicit cytokine release syndrome, and see how sensitive is this device and platform for doing what it's supposed to be doing. And if validated in that sort of phase 2 setting, I think that would be the time I would be very confident to propose to, our scientific societies and also to large hospital systems to consider taking CAR T therapies to the outpatient setting in tandem with a wearable device for close monitoring. So that's how I envision things to change in the future. I would want to validate this in a larger study setting.
Well, what do you think, Dr. Zaradka? What's a research question you would like to answer next?
So many, but what I'll start with is looking at how personalized alarm thresholds and the baseline data change as we move to that outpatient setting. So taking the work we did and to Shree's point, looking at sort of a multi-institutional setting, but also in a different context while patients are home, what is their temperatures looking like? What are their vital sign variations? as a first step to ensure that the work that was done in the inpatient setting is translatable and translational to the outpatient setting. I think that there's a lot of opportunity and work to be done for the early detection / prediction of CRS. Again, forecasting comes to mind in terms of vitals. How far out into the future can we guess what someone's vitals are going to be, and how does that build in sort of more of a buffer into enabling clinicians to reach out and action some sort of care to patients who are highly likely or identified as highly likely at risk of developing CRS in ideally a pretty large window of time, right? The more time you have, the more time you have to change something. So I think that those would be the top two questions I had for this particular work.
Awesome. Definitely a lot of important and interesting questions for the both of you to tackle in your future work. That is all of our time for questions today. If we did not get to yours, we thank you for submitting it. We will reach out to you personally to make sure you get a satisfying answer to that. And thank you all for attending this Fierce Pharma webinar. Thank you to both Dr. Rajiv and Dr. Zradka for sharing about this fascinating and exciting research. A recorded version of this webinar will be available for you to access within 24 hours. You can use that same link that you used to join this one. Thank you again for joining, and we look forward to seeing you at future events. Have a nice day, everybody.